So I'm really pleased to introduce Kelsey Wilson. Kelsey is a senior in biology. She's really interested in natural sciences and conservation, and she really was interested in doing some field work. And so last spring, Kelsey and I discussed the fact that I love working in rivers and being outdoors in the summer, working in a beautiful river, the Cahaba River, which is, runs through sort of the center of the state into Birmingham. It's something that is really great fun, and it's a lot of work, but really neat, exciting work to understand some fairly important species of concern that are freshwater snails. And many of us are not familiar with those animals, but they tend to dominate our river systems as far as how many they are in our creeks. If you look down, they look like they're covered with rocks, and those are all snails. And so we had a funded project to understand what's going on with this federally listed snail, which means if you go and pick them up and take them home, you can get fined. So that's the moral of the story. And because of that, the Fish and Wildlife Service, who regulates endangered species, is very interested in understanding if this one federally listed snail that's a high conservation concern could even become more conservation concern if it continues to decline in its populations. So a lot of what Kelsey's work is about is understanding sort of if these snails are going to be okay by looking at a bunch of different things in the river. And so she did a fantastic job, and we had a great time, and then she had to look at all the data. So that was the part that wasn't as much fun, but something that she did a great job with. So this is Kelsey, and she's going to talk about her work in the Cahaba River.
Okay. I'm Kelsey, and the title of it is Habitat Use and Relative Densities of the Federally Threatened Brown Rock Snail, Leptopsis ampla, in the Cahaba River. So this snail is called the brown rock snail because it is literally like a little rock. It looks like a little rock. Like when you pick it up, you have to really, you know, sift through all the pebbles to see it. What we did, the brown rock snail is only found in this part of the Cahaba River. And what we did is we went to 2 locations at the very top of where it's found and at the very bottom of where it's found. We did 2 main stem locations and then 2 tributaries that runs into the main part of the river. So we did, this is the farthest south Cahaba River at Centerville, and then we did Cahaba River at Helena, and then these are the 2 tributaries, Schultz Creek and Shades Creek, and they look very similar. But what we did at each stream, you can see right here, we mapped it out into 10 quadrants so that in the middle of each one, and it's on the shoal line. That's where, from previous research, that's where they're majority found in the shoal line. So that's where we mapped it out at, and we measured each in 1 quadrant. And also we had to make sure that it was fast flowing because we used an instrument called a server, and it's right here. And what you do is you put it on the bed of the stream where it's fast flowing, and there's like a little bag connected to it on the end where the water will flow through. And you kind of, you know, you can like kick the ground or like, you know, put all the substrate samples inside of it to like, that's your sample of that little square. So we did that 10 times. And then what we do, we get a whole big bag of that out of the sampler and dump it into like a little box and sift through it and see all the snails that we found. And in total, we found 6 different snail species. We also measured the habitat conditions at each of the sites, including the depth, velocity, the particle size of the substrate, and percent vegetation. So, this vegetation, and sometimes there'll be some vegetation in each quadrat, and we measured to see how much of it was in that one site. And for the results, we determined that most of them were found on bedrock. And here's bedrock right here, and here's just like a bunch of snails that are on the bedrock. The lowest densities of snails were found in the main stem sites. See, these are the main stem, the Helena and the Centerville, and you can see that the densities are lower. The blue is total number of snails found, and then the orange is the Leptopsis ampli that we were trying to study. Schultz Creek has had a large portion of bedrock, and it also has the largest densities of snails. So this is Schultz, and you can see that there were a lot of snails, and it also had their largest portion of bedrock, so that goes with our hypothesis. And all of the water chemistry was relatively the same throughout each site, so that really didn't affect anything. Based on these results, population of Leptopsis ampli seemed to be more vulnerable in the main stem sites due to overall low population densities. And so we have mining, urbanization, and agriculture land use is the reason for these snails being vulnerable in those sites, just because of all the pollution from that. And ensuring that bedrock outcrops are maintained and not filled by sediments is critical for these snails to survive, because that is their habitat of choice. And some of these sites, we did do this last summer, and that's in the summer is really when the water's at the lowest point, but there was some of the sites were still kind of too high to measure everything, so really going back and when the water is lower and measuring all that you can find would be the best at determining this, because it was kind of hard to measure all of them if the water is like really fast flowing, and you can't really get your samples. But understanding the relative population densities and habitat use of these snails is important for conservation. And also, we included the Alabama seal because it's the only one where it has rivers on it. So that just shows the importance of rivers and how pollution is like affecting it. And the Alabama is considered to be a global hotspot for freshwater biodiversity. So it's really important to understand the problems with different pollutions and the effect that it's having on different species, and also on the specific snail, Leptopsis ampli. Okay.